Welcome to plant developmental biology. So in last class we started studying root development, so we will continue that. So this slide you would have already seen in the previous lectures. So this is just to remind that there are three major steps which takes place during the process of root development: the root apical meristem, tissue patterning and root branching, which is basically lateral root development. So in last class what we discussed more towards how root apical meristem is maintained. So this is very important to produce or to generate a large number of stem cells. So this is just this picture you would have already seen in the last class, but this is just to recap you before going for the transition. So it is very important to have a group of cells which should always divide and they give rise to the cells which is required for the process of differentiation. And the first and very important thing what happens for the maintaining root apical meristem is positioning a stem cell niche. So this is the reason which is stem cell niche, which means that the cells which are present in this domain, they are stem cells, they have stem cell property, they can divide and they can provide cells for later stage of differentiation, and their position is relative with the QC. So the cells which are in direct contact with the QC, they remains as a stem cells. But next step what happens is the differentiation. So what happens that these initial cells, they depending on their position, they take their daughter cells after the cell division take different identity. So once these cells divide, what happens, let us assume if this is the cell, if these cells divide here, so there are two daughter cells. The daughter cells which is towards the QC, it remains as a stem cells or as initial, but the daughter cell which is away, it enters in the process of differentiation. And just now I said that the fate of these cells, the differentiation program in these cells will depends on the relative position of the cells. So for example, if you look here, so there are few things which I just want to remind. This is the initial which is called CEI, which is initial for cortex and endodermis. So you can see here both the layers cortex and endodermis, they are originated from the single cells. Similarly, this initial is for epidermis and lateral root cap. How this happens we will see later. These initials are for columella cells, they give rise to columella, and these are the vascular cells initials, they give rise to vascular tissues. And once this process of differentiation starts, one thing is very important. If you take this seed, if you take any plant and germinate it at very early stage, as you can see this is at the embryogenic state, the pattern is defined, but at early stage maybe the rate of cell division is slightly more, relative rate of cell division is more than the cell differentiation, but at certain time point there has to be a balance between rate of cell division and rate of cell differentiation. This is extremely important to ensure that a proper amount of meristematic cells are maintained, at the same time sufficient amount of cells required for the differentiation should be provided. One and very important step of differentiation is asymmetric cell division. You can say this is the first step in many of the differentiation program in the life cycle of plant development. If you recall your embryogenesis, so even the first cell division of zygote is asymmetric in nature. Asymmetric means producing two daughter cells which are not exactly similar. So if you look here again, this is the same picture, but what you can see, so we can see that these are the initials which is for cortex and endodermis. And so cortex and endodermis, if you look the first step of differentiation, so after this cell division, so this cell division will generate two cells. These cells which is now in contact with QC will remain as CEI, but these cells will undergo the process of differentiation. And what happens once the differentiation process starts, it undergo another round of cell division. This is the cell division, and now this cell division is different in nature. So, there is a reorientation of the plane of cell division. So, this cell division, if it is periclinal, this is called anticlinal, and this cell division essentially generates two layers from here onwards. One layer which is going to give endodermis, as you can see here, and the outer layer which is going to make the cortex. Similarly, if you look the initial, this initial is epidermis and lateral root cap initials. These initials again undergo first round of cell division. After cell division, the inner cells, the basic and inner cells which are again in contact or which is a part of the stem cell niche will remain as a stem cells, but other inner layers, this start another cell division and give rise to epidermis, whereas outer cells, it will undergo the process of a specific cell differentiation program to give rise lateral root cap. This is important. Third, initials, if you look, these are the columella initial cells. These columella initial cells again undergo the process of cell division, and they remain the stem cells property in the cells which are just below the QC, but the cells which are distal from the QC, they undergo the process of columella specific differentiation program. This is what happens in the vascular initials. Vascular initials, based on their position, you can say they can be pericycle initials, they can be protoxylem initials, metaxylem initials, and procambium initials or phloem initials. So, in all this case, what you see that the process of differentiation start at the first asymmetric cell division itself. Then it undergo lot of programming, lot of cell division and cell differentiation programming together, and lot of genes which are getting regulated or which are getting activated, they are regulating this process. So, what we will do, we will take few of the example and we will try to discuss how this happens. So, if you take the differentiation of cortex and endodermis, and what happens that just now we said that cortex endodermis initial cells, it undergo the process of asymmetric cell division, but what are the regulators? So, if you recall your previous class, you remember that these two proteins are very important, short root protein and scarecrow protein. They help in regulating not only stem cell maintenance, but they also help in giving a specific identity to endodermis. And what happens that this scarecrow protein, it has been found to physically interact with a retinoblastoma like protein. And this, if you look, the scarecrow protein is also interacting with short root and interacting with RBR, but their interaction is totally different. And these three important residues ACA, it is important for interaction of short root protein with RBR. If you mutate these residues at the place of these residues, so this is actually mutant. If you mutate it, what will happen that the SCR will lose interaction with RBR, but interaction with short root protein is not disrupted. And how this regulates, we are going to look here. So, this is a schematic diagram which we have already discussed. So, if you look this picture, what happens that here you have scarecrow promoter driving scarecrow YP fusion protein in scarecrow 4 mutant background. So, this is a close to basically wild type. You have scarecrow mutant, but this mutant phenotype is getting complemented by YP fused scarecrow protein. But if you mutate the interaction between SCR and RBR, what happens that you can start looking some phenotype. And one phenotype you can see here is that there is a extra layer formation. So, normally in wild type from CEI only 2 layers are formed, and in these 2 layers the outer layers always remains as a cortex and inner layer differentiate into endodermis. But here if you look in this disrupted interaction mutants, what is happening, this is more clear on the upper side. If you look, there is, so this is wild type, you have only one cortex or equivalent wild type, and you have only one endodermis. But when you mutate these residue, when you disrupt this interaction between SCR and RBR, you can see that between cortex and endodermis there is extra layer. So, this tells that this interaction between SCR and RBR is very important in regulating cell division pattern. Another important thing what you will look here, that this is one very interesting gene which is called cyclin D6 gene, and if you look its promoter fusion construct, this gene very specifically expressed in the daughter cells of CEI. So, when CEI undergo the first round of cell division, you can see these genes are expressed, this cyclin D gene is expressed, but immediately after that its expression is totally disappearing. So, which means that there is a possibility that the SCR, RBR or these genetic regulators are activating expression of this, which you will look later on in detail, but this might be interesting, because when you disrupt this interaction between SCR and RBR, what you see that there is a extra layer, and more important that the expression of cyclin D6 expands. So, it expands in both the layers. So, this tells that this interaction is not only important in maintaining the number of cell layers, but it is also important in restricting or in regulating the expression domain of cyclin D. Another important thing, if you look here, so this is plant treated with auxin, or you can increase the auxin level. When you increase the auxin level, auxin is a hormone, plant hormone. I have already told in previous class that auxin is very important. It regulates lot of process, and here also particularly in roots, auxin has a very important role to play. And what happens if you induce these plants with auxin, you can see there are extra layers, and these extra layers have induced expression of cyclin D. But important thing here is that in this extra layer, out of these extra layers, your endodermis is only the innermost layer, as you can see from the localization pattern of short root protein, which is also always in the endodermis. Another important thing here, if you look, so this is short root protein complemented with short root GR fusion. So, if you recall one of our class, we have discussed that glucocorticoid receptor fusion to a transcription factor basically make it inducible in a way that when you treat with dexamethasone, then and only then this protein will enter in the nucleus and do its function. But if you do not induce, then the protein is there, but protein cannot go inside the nucleus, and since it is a transcription factor, and if it does not enter in the nucleus, it will not perform the function. So, what happens here? So, if you look here, so in short root mutant, since this mutant we are not treating with dexamethasone, that is why you do not see the complementation. So, this is a kind of typical short root protein mutant, and you do not see induction of expression of cyclin D gene. But when you treat with dexamethasone, dexamethasone is basically allowing short root protein to enter inside the cell, and when short root protein is entering inside the cell, you can see cyclin D expression starts, which means that short root protein is activating expression of cyclin D. And when you treat dexamethasone plus auxin, then you can see that it is mimicking this phenotype where you have extra layers, and extra layers are basically increasing having more or expanded expression of cyclin D. So, this all together suggests that short root scarecrow is important, and it is working to activate auxin dependent activation of cyclin D proteins, and SCR's interaction with RBR is also, all these regulatory networks is very important in regulating the asymmetric cell division of CEI and eventually a layer of endodermis and cortex formation. So, if I summarize whatever we have discussed here, so there are two things to remember. One is that this short root protein, which basically in some of the future class you will know how exactly it moves, but short root protein, it moves from one cell to another cell, and this movement essentially generate a kind of gradient. So, you can see the cells, because you know short root proteins are produced in the vascular tissue, the cells which is very close to vascular tissue it is having more short root, and then similarly the auxin signaling pathway. So, what happens in the auxin signaling pathway? Auxin is synthesized in usually highly dividing cells or in the young tissues, and then there is a well established system that the auxin can be transported in a very specific manner. So, there is a process called polar auxin transport. So, auxin can be synthesized somewhere, but it can be transported to different places, and this transportation basically ensures auxin concentration. And what happens that this basically generates a kind of gradient in the auxin concentration. It generates maximum amount of auxin in some tissues, minimum amount of auxin in another tissue, and auxin works like a morphogen. So, if you have high amount of auxin, it can induce a very specific genetic program. At low amount of auxin, it can perform a different function. So, the combination of all these kind of things, if you look here, so what happens in the position A? So, this is your position A is basically CEI. In CEI, we have high amount of auxin, and then what happens that short root and scarecrow activate cyclin D in auxin dependent manner, and this process essentially induce asymmetric cell division. And after asymmetric cell division, if you look in the inner cells, what happens? The domain of short root and scarecrow is restricted only to the endodermis cells, and how it is restricted is another different regulation which we are not going to talk here. But what happens that in inner cells, since auxin level is going down, so you have short root scarecrow mediated cell fed determination of the endodermis. But overall what happens, the combination of auxin, short root, scarecrow, RBR basically ensures the first or the asymmetric cell division in CEI and then eventual process of layer differentiation. Similarly, similar kind of cell division is also very important in differentiation of epidermis and lateral root cap. So, if you look this is basically epidermis and lateral root cap. What happens that the initials first undergo the process of pericycle cell division, and then it undergo the process of anticlinal cell division, and this essentially results in formation of epidermis and lateral root cap. So, the origin of both the layers are from the same initial. And there are two transcription factor, both are NAC domain containing transcription factor. One is called phage, another is called basically SMB, which is sombrero. And what happens that in the mutant background, what you can see here, the frequency of periclinal cell division is significantly decreased. So, this is the wild type label, this is phage mutant, and this is SMB mutant. So, which means that both are very important for making periclinal cell division. And if you look in the mature root, what happens that in wild type, and this has a eventual effect on the lateral root cap development. As you can see here that in wild type there are 4 layers of the lateral root cap, but when you have a phage mutant you have only 3, whereas in SMB mutants the number of cap layers are increased. So, basically they are regulating lateral root cap development in a different manner. If you look here how columella initials undergo the process of differentiation, the same regulators has been shown to play very important role. So, if you look just the expression pattern of phage and as SMB protein, what happens in the during embryogenesis, the phage start expressing here. At that stage SMB was not activated, but slightly later stage SMB is getting activated. And when you overexpress basically this phage protein, what happens in endodermis. So, you are using scarecrow protein. So, you are overexpressing phage protein, and what you can see there is additional round of periclinal cell division. So, this basically supports the even previous observation that phage is important for periclinal cell division. In mutant that cell division is decreased, but when you overexpress there is additional cell division takes place. But if you look the expression pattern and their regulatory network, it is very important. So, if you look this phage is not expressed in the QC, but it express in the initials, which is columella initials, and then this expression, when these columella initials undergo the process of cell division, what happens after this cell division, the expression of phase remains in the lower cells, but it totally and immediately very quickly disappears from the initials which are close to the QC, and SMB induction starts only in the lower layer, which is your initial daughter cells of the initials. So, if you look all these expression pattern and mutant phenotype, if you combine, there is a regulatory mechanism. What happens that at the very early stage, phase is getting activated, in these cells, then these initial cells undergo the process of cell division. When cell division occurs in the lower cells, phase basically activate the expression of SMB, but this SMB, once SMB is activated, it immediately repress the phase expression, and this how there is a differential expression pattern, and this basically regulatory networks ensures a proper stem cell identity for the columella stem cells and the distal cells to enter in the process of differentiation, proper differentiation program. If you look more detailed how auxin regulates root cap formation, there are other evidence. So, basically these are auxin response factors. So, auxin response factor, a class of transcription factor which basically regulates the process downstream of auxin signaling. And what happens that if you mutate auxin response factor, in single mutant you do not see much phenotype, but when you make a double mutant of auxin response factor 10 and 16, what you see that there is a defect in the root growth. And if you look their basically columella, what happens in wild type, you have maturate or differentiated columella, but in this mutant background most of the columella cells are not differentiated. So, there is a problem in the differentiation, they are more kind of dividing, and layers, number of layers are also increased. Another important thing is that this auxin response factor 10 and 16, they are regulated by microRNA 160. So, microRNA, you know that they are the small RNA, and microRNA, what happens that if they target a gene, they silence its expression. So, what happens, this you can see, if you overexpress microRNA, basically this gives a phenotype very similar to the loss of function of the auxin response factors. So, when you overexpress miRNA, there is more microRNA, more down regulation of auxin response factor, which means that it will have a similar phenotype as the auxin response factor mutant. But this is interesting, if you, so basically microRNA, what happens, these are just 21 to 24 nucleotide molecules, and they have some complementarity sequence with the target, and if you mutate this target binding sequences, this overexpression of microRNA, and then you complement with the mutant version of auxin response factor, you do not see the phenotype, which suggests that the microRNA is regulating auxin response factor 10 and 16 through the binding, and this regulation is basically important in regulating columella cell differentiation or root cap formation. Similar kind of phenotype you can see here. So, if you have wild type, this is the differentiation pattern. If you treat with auxin, so here there was loss of auxin, now we are having a gain of auxin. When you have more auxin, you can see the differentiation, even stem cell properties are lost. You have this YUC mutant is basically defect in auxin biosynthesis, internal biosynthesis of auxin is reduced. So, basically this is low amount of auxin, you can see 2 layer of stem cells. Similarly, if you look this mutant background, and if you look the complementation, or if you make a double mutant, triple mutant, auxin response factor 10, 16, if you combine with the WOX5, all these kind of things, you can see here that these auxin response factor, microRNA, auxin, they all are working through the WOX5 pathway, which means that the combination is not basically helping in detail. So, there are some more important mutants which are playing, maybe are the genes which are regulating later stage of root cap formation. The final stage of root cap formation basically is programmed cell death, because by the process of programmed cell death, these caps are basically removed at a certain frequency. But if you look this mutants background, what happens that this lateral root caps are not getting removed. So, these are basically process of root cap maturation, this is also important. And there are some another transcription factor has been identified which regulate this process. So, if I combine everything together and if we look in a overall regulation of the transition between cell division and cell differentiation. So, what happens that auxin is regulating AR of 10, 16 and these are working through the WOX5 pathway to restrict or to regulate the stem cell niche and allow a proper differentiation program. Here if you look layer wise how regulation is working. So, this is your QC, this is C1, C2, C3, C4, C5 and this is here. So, what happened in the QC? In QC basically there is a less division, less differentiation, or you can say QC is no division, no differentiation. So, but WOX5 is present, but if you come one layer down, this layer is basically stem cells, stem cell has to be divided. So, here division is dominating over the differentiation and what happens, WOX5 is very active, RBR is active and phase is active. But in other layer C2 to C5 layer what happens, AR of 10, AR of 16, SMB, phase and RBR they basically interact and they basically shift the transition from cell division mode to cell differentiation mode. And if you come in the last layer which is called terminal differentiation, the final layer differentiation, there is another gene which is called BRN1 and BRN2 and assembly, they basically initiate or activate the process of terminal differentiation. And that is why if you do not have BRN1 and BRN2, if you remember this, the terminal differentiation is defective and that is why you can see multiple layer of lateral root cap formation. So, we will stop here and in next class we will discuss vascular tissue development. Thank you very much.